Now, I would like to continue with the introduction. Certainly, all the success is due to researchers who build up the field in different areas. And I would like to introduce the keynote speaker, Chad Mirkin. A key achievement at NNI has been a new generation of researchers and innovators. Professor Chad Mirkin is on the top of this wave as a leading researcher, educator, and entrepreneur advancing nanotechnology. I met Chad in the 1990s as a brilliant young chemist at Northwestern University. Chad has embraced the NNI vision and goals from the beginning. He has pioneered important discoveries and has been chief developer and founder of multiple companies. He has become a singularity in nanotechnology community with about 900 papers and 250 awards. And he is member of all 3 US National Academies of Science, Engineering, and Medicine. He is a George B. Ratman Professor of Chemistry at Northwestern University, and he is a director of the International Institute of Nanotechnology in Chicago. Chad has been an outstanding science citizen, frequently testifying and lecturing about nanotechnology and related fields. He has been a spokesman for the community, serving PCAST and many other organizations. In a recent article in Scientific American, Chad wrote, "The NNI promised a lot. It has delivered more," which is also the way his stellar professional career may be described. Chad, we are eager to listen to your perspective.
Well, fantastic. It's a really nice introduction. Really great to be here to celebrate this big event. And I do want to mention up front, I am a huge believer in the NNI. I remember when it started, a lot of people thought in science that this was a gimmick, a way to kind of rephrase what we already do in science, and they couldn't have been more wrong. And many people in the room, I think, realize that. Mike in particular, OSTP, Tom Khalil and company, they realized that this was a way to really change the trajectory of science in engineering and medicine, frankly, in many different ways. And those that rallied around it, I think ultimately did quite well. And if you look at how the US made bets on it and the output of it, we did quite well. Turns out when I was at Northwestern, I was a young professor and I decided I was going to make this my area. I was very excited about the science of miniaturization. And we began to build the first International Institute for Nanotechnology at Northwestern. In fact, we were breaking ground 2 years before the NNI. And so we were opening up the first federally funded International Institute for Nanotechnology at Northwestern right when President Clinton announces the NNI. What a great place to be in terms of starting your career. And so we began to take it very seriously and build teams that really thought about capitalizing on actually, where's my slide turner? Oh, here, right here. Capitalizing on the great opportunities that were put forth. And you know, people were talking in hyperbolic terms about what this was going to do. To me, the exciting thing was that this was a field about changing everything. And we thought about that in the same way. And so today, we have an institute that focuses on really many of the key issues in nanotech. It's not centered on just one topic. The first and most important thing is in the development of tools that allow you to miniaturize and study miniaturized architectures. That's where all the action is, right? And you have to remember, many of these tools didn't exist at the time. To me, this is so exciting because science in general is driven by tool development. When you change analytical benchmarks, you change science. And so developing new capabilities that allowed you to manipulate matter on this scale made a huge difference in terms of almost all fields. And so a big part of what we do focuses on creating those teams to create cutting edge tools that drive the field. But once we have those tools and can make new materials on this scale, we then focus on utilizing them to solve environmental solutions, food and water safety, energy solutions, and redefine how we actually do medicine. And so what I thought I would do today is take a couple examples from my laboratories at Northwestern. We have many, and talk a little bit about how we think about them and what they've ultimately led to. And I want to start with this first premise, which I think is the fundamental tenet of nanotechnology, which is the fastest route to new materials is through miniaturization. That's what was so exciting. That's what Mike Rocco recognized from the beginning when he began to build teams of people throughout government and also throughout academia and industry to realize, hey, this is going to change everything. Everything we take and miniaturize will be different. Chemical components that are reassembled on the nanoscale are different, not because they're small, actually because they're large. I think people lose that. This is really the length scale in many respects of biology. It's a length scale that's considered pretty large from a chemist's perspective. But taking a structure and putting it together from multiple components can yield a material that has properties that we've never seen before. And so the two examples I'm going to share with you are signature discoveries out of our lab. One is the spherical nucleic acid, and the other is the development of nanoprinting tools, tools that allow you to print with all forms of matter on the nanoscale routinely, not in a fab, but at the point of use. These were actually coined as desktop fabs when they were first invented. You'll see how far they've come in modern days. These discoveries have led now to a lot of technologies that are impacting many people in the room and people around the world. Medical diagnostic tools that are now used in over half the world's top hospitals. New therapeutics, shots on goal in terms of developing therapies that can treat some of the most debilitating diseases. Desktop fab capabilities that allow you to miniaturize structures routinely, not again at a remote site, but at the point of use. These are workhorse tools used throughout academic labs and industry. And those tools in recent days have transitioned into the world's most parallel synthesis tools. They are catalyzing the way we synthesize materials, changing the way we synthesize materials, enabling us to make more materials than we've cumulatively made to date in a single experiment, which is pretty exciting if you think about what's going to come out of that. And then finally, something we could have never anticipated, spanning 15 orders of magnitude, the pursuit of these led to other things like the development of the world's highest throughput 3D printing capabilities. Again, going from the very tiny to the very large. Okay, let's start with the spherical nucleic acid. This is a structure that we invented or developed back in the mid-90s. We reported in a paper in Nature. To this day is the most cited paper in Nature in all fields, I believe. I guess 8,300 citations and growing, because it was a new concept. The idea was that you could take two materials, disparate materials, a nanoparticle and DNA, and interface them through chemistry and concepts from nanotechnology. And that led to initially what we called spherical nucleic acid gold nanoparticle conjugates. You can see in the upper right or the middle right panel an EM image of the particles where you clearly see the gold nanoparticle core and the shell of nucleic acid surrounding it. These are structures that have no natural equivalent. They're made purely through chemistry and concepts from nanotechnology. There are linear forms of DNA, there are circular forms of DNA, there are folded forms, there are not spherical forms in nature. Yet these structures interact with living systems completely differently than those entities. And those differences, some of which are labeled below, have led to unique science, but also unique technological capabilities. And I want to talk a little bit about some of the translational activities, because I think that's really important to look at how far these types of things have come. But I point you to the one experiment. This is an AHA experiment. It took us about a decade to run it because you're taught. Conventional wisdom is that this shouldn't work, that linear nucleic acids will not enter cells, human cells. These are epithelial cells, you can see. Linear DNA with a fluorophore, put it in cell culture, the cells reject it. Take that same DNA and rearrange it on the nanoscale into this new spherical form. Now it not only goes in, it goes in better than anything known to scientists. Actively taken up, rapidly internalized. The reason that's an aha moment is it's odd. You say, why does that happen? It turns out it's a scavenger receptor mediated process. We've actually studied it in great detail, but it opens up a new way of thinking. Now we can move large amounts of nucleic acids into cells simply by controlling architecture on the nanoscale. And that opens up the ability to develop a lot of new medical diagnostic tools and a lot of therapeutic shots on goal. So that's what we did. And so this sent us on an odyssey over the next 20 years, which continues today, where we develop in vitro diagnostic tools that are now used for things like COVID testing, among other types of diseases. New ways of measuring the intracellular contents of cells so that we can track cells and differentiate them based upon those contents. And a completely new way of thinking about medicine development. I call it structural nanotechnology or structural nanomedicine, where you can begin to take many components and put them together in unique structures that lead to then efficacious therapeutics or not. Led to a whole series of companies that many of which were taken public, some sold, that continue to this day to develop new capabilities and will continue for many decades to follow. So let me give you one example. This is the Virgin system. It was first commercialized by our first company we started called Nanosphere. The basic idea was to create a gene chip type of assay where you have different quadrants of DNA on a chip that can capture different signatures of disease, nucleic acid signatures of disease. If a particular signature is present, it'll go to the right spot on the chip. It can be sandwiched with a nanoparticle probe, in this case, a spherical nucleic acid that actually can sequester lower concentrations of that particular target. So you get higher sensitivity because of that. It has higher binding affinity than normal nucleic acid type probes. And that gives you one level of signal. The gold, if nano gold is not gold in color, nano gold is red in color. Again, a consequence of miniaturization. That gives you one level of signal, but if you flow photographic developing solution over this chip, that's silver 1 and hydroquinone for the aficionados. Anywhere where there's nano gold, you facilitate catalytic deposition of that silver, and now you can amplify the signal by a factor of 10 to the 5 in a few minutes. And so now you have one of the most sensitive detection systems out there for nucleic acid signatures of disease. And in this particular case, you can use a colorimetric reader, and you guys can read the results. This is a infectious disease assay, HIV, Ebola virus, smallpox, hepatitis B. Experiments done 6 times over for each disease target. And you can go clearly through that and see the positives and the negatives just with the naked eye. If you want to quantify, you use a reader like the Virgin system that was developed for this. And that technology, which took about, the first paper was in Science magazine in 2000, I believe. It took about 15 years before where it is today, now used in over half the world's top hospitals. A backbone of Luminex that bought Nanosphere, and then Diasorin, which bought Luminex. But we wanted to do more. So these structures that had these fantastic properties, how do we take advantage of them? Well, we thought maybe we could use these to create probes that could measure intracellular contents. So we developed a construct called a nanoflare, which is a spherical nucleic acid with a gold core, and it is a short oligonucleotide with a fluorophore hybridized to it. So the fluorophore is near the gold and is spectroscopically silent. These structures are designed to go into cells like normal spherical nucleic acids. And if there's an elevated level of a target mRNA fragment, they will bind to it and peel off the flare and turn on fluorescence. And so now you have the first way to measure the intracellular mRNA content of live cells. So I can use that to look for the proverbial, a needle in a haystack. Let's say I want to look for a circulating tumor cell in the presence of healthy cells. I can fire this through a flow cytometer and simply look at the fluorescence and set it at certain thresholds, taking out those cells in live form. And I get them in live form, so then I can culture them, and I can begin to look at, for example, how they respond to different types of experimental therapeutics. So this is a pretty amazing capability that's now used for that type of exercise, isolating circulating tumor cells, high throughput drug screening, for example, looking at how different markers elevate different mRNA in live cells. And even in live animals, you can use these types of entities to measure mRNA content. And some researchers in Singapore were utilizing them to try to identify the origins of keloid formation, these terrible scars that form. They're very prevalent in the Asian population. They were concerned about that and showed for the first time in rabbits that they could actually measure mRNA content and correlate that with keloid formation and hopefully develop therapeutics that minimize that. From a therapy standpoint, we've already heard, nanotechnology is impacting the development of vaccines. I'm a big believer in that. That's just scratching the surface, though. That's, in fact, a really crude use of nanotechnology. It's basically a small liposome that encapsulates RNA and delivers them all in one construct. But it's an important step in the right direction, because think about vaccine development. Okay, this is a field where we focus really on two things: something to stimulate the immune system, an adjuvant, and something to train the immune system, an antigen. We mix them up, we inject, we hope they work. If they work, we run with it. If not, we say, well, we didn't have a good adjuvant or antigen. I actually question whether that's correct or not. We've shown that if we compartmentalize these, and in the case of mRNA vaccines, get your body, your cells to produce the antigen, we can take a step in the right direction. And that was a really, really important step, and there are advantages associated with that. But we think you can do more. And so one of the things that we're thinking about is how do we begin to use nanotechnology to structure the antigen and adjuvant in a way that it can maximize the effectiveness of a vaccine. I don't want to take you through the entire way vaccines are developed, but I do want to point you to the fact that when we deliver these types of constructs, we want them to go into a single cell, so compartmentalizing them is important. Naive dendritic cells. And then we want to control two signals: the stimulation of those cells and the processing of the antigen on the surface of those cells. Because what happens then is T cells dock on these and then are matured and are trained then to go out and only find cells, in the case of a cancer vaccine, cells that present that particular antigen and kill those. That's how you get the selectivity. But think about it. This is crazy that we hope these two components will somehow magically get to where they need to go and work in the most optimized form simply by delivering them as an unstructured solution. But that's the way we do things. So one of the things we've been asking is, can we actually develop a new class of structural nanomedicines? Can we take a spherical nucleic acid and ask the question, can that be used as an adjuvant to improve the effectiveness of different types of therapies? Can we use that as a basis for developing new types of vaccines? And so there's a drug called Cavro, where we've actually tested this, not just in animals, but in humans. For, again, the aficionados, it's a TLR9 stimulator. That's the immune system stimulator agent. And we ran a clinical trial where we asked the question, can these drugs work in combination with these powerful drugs called checkpoint inhibitors? Pembro, Keytruda, all these types of miracle drugs that work really, really well for a small portion of the population, but not the entire population. And one of the theories behind why they don't work so well for the larger part of the population is that you need to have the immune system upregulated enough to work in concert with these. So here's an example of where we can begin to use these types of constructs and ask the question, does structure matter, and can we stimulate the immune system in combination with these? And we did this with two really orphan diseases, but important diseases: squamous cell carcinoma and also Merkel cell carcinoma. And all of these patients had failed all of their therapies prior to this therapy, including treatment with just checkpoint inhibitors. So we gave them this drug along with Pembro, and remarkably, we saw tumor shrinkage in 37% of the patients, and we saw a complete responder like the one shown in this slide right here, which to me is just striking. I have a colleague who once said, my goal in life is I'd like to be able to cure at least one person of cancer. I think we can say that now. In this case, a 92-year-old has got Merkel cell carcinoma, tumors riddled throughout their arms, pre and post-treatment with the drug, and you can see for yourself the dramatic effect it's had. And again, this person did not respond to those therapies. So this is really exciting. It's the first example that shows if I restructure nucleic acids in this form, I can get a construct that will work in combination with at least checkpoint inhibitors to create a significant and positive outcome. But as I said, we want to do more. So one of the things we're asking is, can we redefine how vaccines are developed? I look at the way we develop vaccines today as what I call the blender approach, the mix, inject, and hope for the best type of approach. Again, not making fun of that, because it's actually worked in many cases, and it's actually a pretty simple way of doing things. But think, we don't develop drugs that way. Think of small molecule drug development. We worry about down to the single atom and single bond, how these types of things are constructed. Yet in vaccine development, we haven't done that. And I think there's an opportunity to do that. So on the right, we're asking the question, can we take nanostructures, rearrange the different components that define a vaccine in different forms, where they're compositionally identical but structurally different, and ask the question, does that make a difference? Does structure matter? And here you can see at least a snapshot of that. I took out a lot of the details in this, but you can see where we look at an animal that has HPV cancer, a human papillomavirus, and we're using an antigen, a DNA adjuvant mixture as the vaccine, the traditional type of vaccine mixture, compared to 3 different structural forms that are compositionally identical but structurally different. One we call the encapsulated, where we put the antigen in the core, anchored, where we put it in between the adjuvant DNA, and hybridized, where we synthesize an oligonucleotide tail and hybridize it to the adjuvant so that they're paired together. And again, you might think this would make a difference, because how these things are processed by the biological milieu, how they generate those signals could differ. Well, look at the results. If you look at the mixture, it has almost no effect. The encapsulated, almost no effect. The anchored's pretty good, that's in purple. Look at the red, that's the hybridized structure. The animals go into complete remission, and they have retained immunity. You can re-challenge them with the cancer and they reject it. You look at the survival curves, shocking. So this is what's made me a big believer in this concept that we've got to redefine how we think about developing vaccines, and we have a big program to do this at Northwestern. Oops, it's not going. There we go. Oh, how do I go back? Yeah, so, and this is not a one-trick pony. I mean, this, you pick the type of cancer, without exception, we can find structures that not necessarily take you to curative, but move you down the ineffective to curative axis. And that's a big deal, because I think it tells everybody we better rethink how we develop these types of, we could be missing the forest for the trees. We could be passing on the right combination of adjuvant and antigen, not because they were the wrong selection, but because they were in the wrong structural form. And so a company called Flashpoint Therapeutics is now trying to commercialize this concept and push it very rapidly through the clinic, and we'll see where that goes. I want to finish then with just an example on the printing side of things, because I think that's equally interesting. Back in the late 90s as well, a lot of action was happening in the 90s. We were just beginning to train ourselves on the commercial tools, AFMs, that were coming online. And I sent some students down to run some experiments, and we discovered something called the capillary effect. That is, when the AFM tip is brought in contact with the surface, water will collect at the point of contact. That water sucks the tip down, is viewed as a problem in AFM. We looked at it as chemists and said, hey, we could use that water as a solvent, and we could put molecules in the tip and move the molecules from the tip to the surface. And if they reacted with the surface, we could create stable ad layers on the surface. And that would give us the ultimate tool for miniaturization. We could begin to miniaturize, look at the consequences of miniaturization across many different materials and length scales routinely. But that was the birth of a whole new way of thinking about nanofabrication. National Geographic picked this as one of the top 100 innovations of ever, which was a bit of an overstatement. So, scientists get a little hyperbolic. I think President Clinton was a little bit in his speech at Caltech, but that's exciting. I kind of discounted it a little bit, but I actually think National Geographic was right. And I'll tell you why I think that was right in a second, because we didn't see how far it was ultimately going to go at the time. It's moved now from this idea of miniaturization and nanofabrication to one of the world's most powerful synthesis tools, and ultimately to the development of tools for high area rapid printing in 3D. So similarly, timeline of development, we are developing this, and we are thinking we're going to use AFMs to build everything. Well, pretty soon you realize this is horribly complicated. AFM has a field of view of 100 by 100 microns. You have to make it more parallel. We built the world's largest cantilever arrays ever built, 55,000 in a square centimeter. They were functional, they were commercial, but they were horribly complicated. And then we got an idea. What if we take the spring defined by a cantilever, AFM arm, the cantilever arm, and put it into the tip? And we could do that by making the tip out of an elastomeric material. And we could take and create a stamp of pyramids and mount that on a transparent flat substrate, insert that into an instrument that can move back and forth and up and down with nanometer scale precision. And now we dramatically simplify and parallelize the capabilities. And that was the birth of polymer pen lithography and the idea of cantilever-free scanning probe printing. If we go back in time, this is what we would have done from the start. Because each of those tips now can be point sources to deliver molecules to a surface, and you can build over many square centimeters very, very rapidly, and it's almost unlimited in terms of scaling. These became commercial tools. A company called TerraPrint that actually started with my son. They sell these all over the world now. They're used primarily for research purposes, but they're also now being used to do something that I think is going to completely transform much of science. And that is to synthesize matter faster than it's ever been synthesized before. So you can use these tips if they have different chemical components on them to now make chips that are about 2 by 2 centimeter with up to 5 billion new materials, all positionally encoded on them. Systematically defined to be different in terms of size and composition, and those represent major shots on goal in terms of new capabilities. New catalysts, for example, that will take CO2 and convert it to high-value products. New catalysts that will split water. New catalysts that will actually drive many of the transformations that are critical to the chemical industry and the oil and gas industry. It also represents a way to create chips that give us big data under highly controlled conditions. And that big data is unprecedentedly large and unprecedentedly high in quality and can be used now to drive the development of AI for materials discovery. And so companies like Toyota have recognized this, among many others, and they've begun to work with us to begin to develop these capabilities. And a company called Mirkin, a startup company we started, is actually poised to really do this in a significant way in the clean energy space. So just to show you quickly what these tools are capable of doing, this is a commercial tool here used for making microfluidics, bioengineering, or lithography. So prototyping for electronics and microfluidics, patterning biomolecules like DNA and proteins, enabling unprecedentedly challenging cell surface type studies, polymer nano printing, and as I said, parallel synthesis. And so if you look at some of the capabilities here, you can see reduced, this is why it is a desktop fab. I can go rapidly make a microelectronic or microfluidic type of architecture. I can study it as a researcher without going to a remote facility, and I can change it on the fly. Anything that can be digitized can be printed with this because it's all direct write. In the middle, you can see how we're building protein patterns that control not only how cells interact with those, but the shapes they adopt and the cytoskeleton formation and how they're stressed. For example, we can begin to probe how stem cells differentiate based upon those stresses. And we've created surfaces that can drive stem cell differentiation down the path of forming a neuron, down the path of forming a bone cell, down the path of forming a fat cell. A pretty interesting capability, all coming from subcellular nanopatterning capabilities. And on the right, you can see what I was talking about, this idea of creating this convergence loop of synthesis, screening, and machine learning capabilities that drive ultimately how we discover new materials. And so I want to give you a number. By next year, we will make over 1 trillion new materials using these types of tools. The world to date, in terms of inorganic materials, has made and characterized less than 1 million. So just think about how profound that is and how important that is in terms of driving scientific development. And just to give you a flavor for what you can do with this, you can find new materials that enable the HER reaction. We found the world record at the time, platinum gold copper catalyst. With the Air Force, we found a new catalyst for generating single-walled carbon nanotubes, gold-3 copper. I point you to that. We find lots of catalysts. What's really important about this is because you can go for so many shots on goal, you find global winners as opposed to local winners. The world today does not work with the best materials for a given process. It works with the best materials that we found at that point in time. And then we build an infrastructure around with it and we're stuck with it. This is going to help avoid that problem. We found a world record for blue emission in terms of halide perovskites, new photocatalysts for environmental degradation or environmental pollutant degradation. And then we are channeling this towards finding a replacement for the OER reaction. The holy grail there is to find a material that replaces iridium. There's not enough iridium on the planet to meet the world's needs. So finding something that works as well as iridium, but without iridium is a big win. I show you one here where we've reduced it by 50%. I can tell you today we've actually eliminated it. That will be coming out very soon. Very exciting discovery. And it's all these. Look at the combinations. You're never going to find these serially. They're very complicated materials, and they're found through this massive capability. Okay, so how do you then go from tiny to large? In developing those nanoprinting tools, we had to create ways of keeping materials from sticking to the tips, and we used a fluorocarbon oil. And we started thinking about this. We said, we don't really need the tips if we're going to print polymeric materials. We can get rid of those tips and we could print large area type structures. So we could put a fluorocarbon oil on the window that we shine light through in a 3D printing experiment. And instead of printing layer by layer on a metal object, we could print continuously. And we could take care of one of the biggest problems in 3D printing. And that is the active removal of heat as the structure is generated. That's the biggest limiter in printing big in 3D printing. These are exothermic reactions. You generate a lot of heat. So we'd flow this fluorocarbon oil over the window where we're printing through, and it would keep the material from sticking onto the window as we print, but we could also circulate it and move it through a chiller and actively remove the heat as it's generated. We could also filter it and bring it back in pristine format. So this can just go continuously, nonstop. And that has become a commercial tool through a company called Azul 3D. And you can see here, these are big structures. I can print you in a couple of hours. This is accelerated so you can see it. It's a propeller, a commercial grade propeller mold, and built routinely with these tools. This is going to change manufacturing, how we produce things in the medical device area, prosthetics, braces, automotive, industrial, sports equipment. You can see in the lower right, that's a pickleball racket, fastest growing sport in America. I'm sure there are avid pickleball players out there. And we've even found out we can print lenses because of the continuous nature of this. The commercial grade lenses for your eyewear and for virtual reality screens. That's a game changer. I mean, think about how much we waste in terms of having inventory. This changes how you think about that. You take a process that just wastes roughly 90% of the puck used to make your lens, where you have to have an enormous inventory, down to not needing that inventory and eliminating a lot of the steps associated with making that eyewear. At least that's the vision going forward. Okay, the last thing I'll mention just quickly is by being part of this, and Mike was so instrumental, and as I said, got us all excited about nanotech and really the importance of working with government and working with industry and all the different parties that matter. I had the good fortune of working for President Obama for 8 years as part of PCAST. And we did a lot of things there, but one of the things I was very excited about was being able to help navigate the NNI and help OSTP and NCO make this an incredibly productive entity. And one of the things you realized with that exercise was just the impact it had, not only on my own personal program, because it did. I mean, it really defined how I think about science and how many people out there think about science, but the infrastructure it built and the lead role it played in terms of driving the development of this field. When we toured not just the US, but all around the world, you could see that people were following the US lead. And the investment, I think, has paid off extraordinarily well. I don't think this is the end. I actually think we're still in the beginning of it. We've just scratched the surface in terms of capabilities. I think the world needs to take it very seriously, and we need to double down on what we've already started. And we've got a good, I think, position to do that, and we'd be crazy to squander it. And you see that in every other country you visit. People have actually followed the lead. They're doing the same types of things. They're about a decade behind us in terms of the good things that were put forth here. I'd hate to see us ultimately lose that moving forward. The other thing that's important are the people. And so the people that have come to the lab, this became a magnet. Fraser Stoddart used to refer to Northwestern as like Paris with the artists in the context of nanotechnology. All the great nanotechnologists were flocking to Northwestern, including him, but also most recently, we just moved Ted Sargent and Shana Kelly, two incredible people from Toronto. This is a magnet that draws talent. Talent drives everything that we do here, and people can't forget that. We have to be the biggest talent magnets out there. And in my case, you can see the different people all over. And these are professors that are running their own programs in nanotechnology and kind of spreading the word all over the place. But again, it all comes back to the US and comes back in large part, in this particular case, to what we've done at Northwestern. So I'm really excited about that. And I just love watching all the things that they do turn into great science projects in their own right. Okay, so I'll finish by just acknowledging the folks. This is my group at Northwestern. I'm not going to go through and mention everybody. Agencies, really everybody throughout the years. This is the most recent ones that have supported what we're doing. Again, the pan-agency participation, really, really important in terms of making this a possibility. Broke down the barriers in terms of not just scientists, engineers, and medical folks working together, but just people that considered themselves in one agency versus the other. It created a tremendous amount of collaboration that I think has led to a lot of good. Thank you very much.
Chad, we have a couple of questions. Do you want to take a minute to answer a couple of questions? Please. And I want to remind everyone, please use Slido NNI 20 to submit your questions. So the first one we have is, what key goal should the field of nanotechnology focus to achieve in the next 5 years? What do you think?
Well, I mean, I think a lot of that depends upon, your perspective as a scientist and what you're ultimately brought. I can tell you what we're trying to do is we're trying to dramatically change the rate at which materials are synthesized, the way in which we can discover new materials that make a difference for almost all applications. We're focused initially on electrocatalysis, but this is, pan science, whether you're talking about optics, metamaterials as an example, or electrocatalysis. The tools will be the same. That's, to me, what's so important here. These nanotech tools that just didn't exist a decade ago are now big enablers that are going to dramatically change the field of chemistry and how we think about synthesizing materials and then ultimately finding materials that matter. And then I think the grand goal ultimately is utilizing that capability to empower AI in a way that it's never been empowered before. I think we're becoming believers in AI. You look at what's happened with AlphaFold. That was a big problem that's solved. If you talk to the biologists, they'll tell you it is a really important tool and one that has changed the game in terms of how they do their science and understand protein structure. The challenge of doing that on the material side is even greater. But I think we have the ability now. AI is almost always limited by access to high quality data. That's what these tools give you. And that's what I'm really excited about.
Thank you. And to tie into that, a nice segue, another question was, with the development of trillion new materials through these advanced techniques, including AI, how do we make the resulting data accessible and searchable for other researchers to find uses for these materials?
It's a good question because there's an interplay between science and what we do at the university and what's done ultimately at companies. I think that take ChatGPT and what's been done there in terms of how it's been made open to other folks to ultimately utilize and explore. There's a model there. I think that you're going to see something similar here. You're going to see machines that are sold or built that are educated with this type of data that'll take us up to a certain level. And then I think you're going to see them more broadly disseminated and people ultimately using them to build off of that. In terms of categorizing the data, that's a bigger challenge. It requires a lot of time, a lot of money, and may, if it becomes important enough, may require a dedicated effort at the government level to fund and orchestrate that to some extent as well.
Thank you. And do you think, yes, we're going to thank you all for your questions. I think we've already eaten into our break time a little bit. So let's thank Chad one more time.